everyone, welcome to the Polymer Science Podcast. I am Alicia Buertes. I have a PhD in Chemistry and Polymer Science. This podcast will be bringing you candid and interesting discussions with hardworking researchers that are contributing to the studies which are relevant to our lives. Hope you can learn something new and that you enjoy the conversations. In today's podcast, we speak to Dr. Zhugen Yang about paper-based devices for detection of Covid-19 in wastewater. Dr. Yang is currently heading the Sensors Lab at Cranfield Water Science Institute. He has recently developed a low-cost paper origami device for rapid diagnosis of infectious diseases in India and has field-tested malaria in Uganda. In the following conversation, Dr. Yang speaks to me about the prospects of this device and how it can contribute to the current pandemic by lowering the pressure on the testing facilities, as well as establishing the presence of the COVID-19 in communities that are at risk. Well, firstly, how is things going there with the lockdown and everything? Are you allowed to work or do you have to stay at home?
Currently, we're mostly working from home.
Oh,
the
yes,
university is closed.
Okay.
But it's another way. Every people just justify ourselves to fit this new situation.
Yes.
So it's okay.
Yeah.
So actually, where are you now? You are in South Africa.
Yes, I'm in South Africa, Cape Town.
Cape Town. All right. Okay.
Are you very busy at the moment, or how's things going? Is it much more?
Yeah, it's a bit busy. We are trying to developing the device and carry out the experiment.
Right.
We because of this particular project, the university just permit my student and I access to the lab and just working on this topic. So a bit busy, yeah. But it's fine.
It's very
Friday,
time sensitive and all that as well. Yeah, you wanted to have you want to put it out into the world as soon as possible. Well, can we just start with the
we should start
first? That you can maybe just for the people who's not familiar with your work and with yourself, maybe just like a quick brief introduction of your name and how long you've been working on this project and what inspired you to start working in this field.
Yeah, so basically my name is Zhugen Yang. I'm currently a lecturer in sensor technology within Water Science Institute at the Cranfield University. I'm a material engineering by training, but I changed my area at my PhD to biomedical engineering. So as a result, my expertise is quite high disciplinary, involve chemistry, material, nanotechnology, medical science, environmental science, and some applications for the sensing technology. So I got my PhD, got my degree, bachelor and a master in China. Then my PhD from the University of Lyon in France. Then I started my career as a postdoc at the University of Cambridge in the UK, and then I got a position from European as a Marie Curie Fellow based at the University of Bath in the UK. At that moment, I started research on the wastewater analysis, but most is focusing on illegal drugs. So that's an amazing project. We're monitoring metabolites of illegal drugs like cocaine or methamphetamine in different European countries. Then that technology was expanded to China, Australia, USA, and across the world. So that's why we have been working on the wastewater analysis for 7 years, I think. So just most recently, for my expertise, I'm working on the development of a range of sensor technology. So this gives my great interest to look at the pathogens and organisms in wastewater in order to understand the community-wide health and infection disease. So because of the outbreak of coronavirus, my attention is just moved to this to trace this carrier of coronavirus in wastewater and also try to understand those outbreak within the community in the population by analyze those coronavirus in wastewater. So that's why we have been working in this area for quite a long time. Just it's a good fit that we using our expertise and technology to address these issues.
Yeah, that's actually
Yeah,
amazing that the technology was already there and established for something completely different, and now it can be used for this. Yeah, it's just
yeah, yeah.
very amazing,
And
actually.
that is one side of about the wastewater analysis. Another side is that actually just did back in my career from University of Bath, I moved to Scotland and joined the University of Glasgow. So then I started working on a paper-based device. So
Hmm.
initially, this device is for the development of biomedical diagnostics. So we developed this technology and then went to India to do a field testing of early diagnosis of infection disease for bovine in a local farm. At the same time, another application is to try to look at the human health within malaria. So my colleagues and I, we deliver this device in Uganda, in Africa, try to test the malaria in the local primary school. We try to do this test within the low resource setting areas because in those areas we don't have really advanced equipment
Hmm.
and like a qPCR or some other facilities to test those infection disease. So that's why I give our another platform to do the field testing of those organisms or infections at the site in a field. So to based on those backgrounds, we propose using these paper-based sensors to rapidly and field testing with coronavirus in order to trace the carrier and understand the cross-infection at the population level and also to predict the outbreak with these ambitious.
Yes.
So that's the basic background of those two stories aligned together.
Yes, yeah, like a very good alignment actually, because now you have a lot of data, or you understand how this device works and what's the fallbacks and the benefits of using it. So for the first question, I actually wanted to know, just for people who don't know what a paper-based device is, what exactly, how does it work, and how is it manufactured?
So this paper device is basically the whole device. We have three sections of the techniques. So first is about the sample preparation. So we use basically using this paper device to uh, lysis, extract nucleic acid or genetic material from different samples like water samples or blood samples. So after this step, we using the folding and unfolding technology, try to drive the liquids in a different panel of the device, and then enabling the proper purification of the genetic materials. So this is basically the first step. We're using this paper device to by folding and unfolding at different time scales to um, lysis and purify DNA samples from the real blood or water samples. So after this step, we design and develop a very sensitive molecular assay, which is called loop-mediated isosomal amplification, or we can call it LAMP. So this technology is similar like qPCR, is quantitative polymerase chain reaction. So they enable amplify the genetic material of virus to be detected at very low levels. So the third step, we're using a visual detection technology, which means that after this amplification, we can directly identify the positive and negative results by your naked eyes. So the whole process involves those steps, but we managed to integrate all of this process in a single device, which achieve the sample in to results out. So in terms of the manufacturing of this paper device, it can be manufactured by a what called a wax printer, which is similar like office printer, but the difference is that we're using the wax as an ink to replace the traditional carbon ink. Yeah, so it's very cheap and easy to manufacture.
Yes.
But based on this wax material, we are able to generate this hydrophilic and hydrophobic channels in order to manipulate these samples of the liquid, but without any power. Unlike traditional microfluidic technology, you need a battery, electricity to drive the power in order to manipulate these different liquids. So that's the basic working principle of this paper device.
Oh, yeah. Okay, awesome. Thank you. Why exactly is paper used?
Yeah, because paper is very cheap first, and also it is easy to store. And from the environmental friendly point, after use, we can just burn out without any pollution to the
Okay.
environment.
Oh, yeah.
And
Yeah, because that's
that's
also the main
new
focus for
reason.
many people, the environmentally friendly devices that can be incinerated or reused again. It's
Yeah.
actually one of my questions for later as well. But I think I'll ask them while we're at it now, do you think these devices can be reusable in some way, or do they simply get destroyed and then
Yeah. Actually, for current design, we don't want to use it for many times, and it's just been single-use device. But it's quite disposable. As I said, we can just simply burn it out.
yeah, but that's good. I think being for one use only, since it's such a highly contagious virus, you'd rather incinerate it than try to reuse it again for another.
Yeah, yeah.
Yeah,
It's also avoid any biological hazard in terms
yeah,
of the residue of these reagents. Yeah,
exactly. Since we're using or we're testing wastewater as well, so we
yeah,
don't want that to get into anything else. So the sanitation in the communities that we have around ourselves isn't always up to standard. So we have wastewater that's flowing into the streets, and people get exposed to that. So many communities actually have sewage that's in and around their house, and they
yeah.
sometimes not even aware of it, that they are drinking wastewater or coming into contact with wastewater. So this is why I also reached out to you, because it's very, very interesting. And I think, especially in South Africa, we have a lot of communities that will benefit from having a paper based device like yours. So I was just wondering, how far do you think the development for this device would be, and for actual use for communities that I spoke of now in South Africa? How far do you think the development process are at the moment to actually use these devices in real life?
Um, so uh, that's a very good question. So actually, what the studio now is we have developed several prototypes of this paper device. As I mentioned earlier, we tested this device for infection disease in India and malaria in Uganda in Africa. We have a couple of the device which is already being approved to be working on this testing.
Okay.
But honestly, for this new target, we are still working to improve or adapting our device to be able to test this COVID-19. There is a couple of difference between our previous development, but it takes some time to prove it works and implement for rapid testing of COVID-19. So one difference is that previously we tested DNA-based pathogens, but currently we need to adapt for RNA testing. This is not difficult, but just we need to take our time
Mm.
to produce this data and prove it works. And
Yeah.
yeah, and secondly, there is some difference in terms of the samples. For example, the RNA is not very stable in the sewage. We try to target with very stable RNA particles in order to prove that this assay still be working. So that's why also pose another question that using laboratory-based method, you need to pay a lot of efforts in order to ensure the stability of these iron particles in sewage. But likewise, if you can test in those iron particle at the site of sample collections, so you perhaps can avoid these issues
Mm.
because you already tested them. You don't worry about the storage and transportation of those sewage samples into the laboratory and then store at -20 or -80 degree for the further analysis.
Mm.
So in terms of the real-time scales of achieving the real practical application, it depends, but it should be not takes too long for us to prove that it can be used at the site of the sample collections. But currently, we are working on this improvement. We have get some data that perhaps it will be working. But in terms of the really, like you mentioned, like a product, like can be translated as a product or for real practical application, I think we need to working with different people who are interested in those areas for the knowledge transfer in order to put them in the real products. Because we are academic, we are research based at the university. So that is another part of partnership work which we need to be working together with the industry partner.
Yes, yeah. No, I think there's a lot of people that would benefit from your product. Also, I wanted to know these devices, have you established how do you make them selective for COVID-19 specifically? What type of loading do you use, if you are allowed to say?
Yeah, sure. That is also very good questions in terms of the analytical performance. For this kind of device, most important parameters is sensitivity and selectivity. So we have several strategies to ensure that this device can selectively and sensitive test of the ion particles or our target of interest. So firstly, we have using this device for proper sample processing. As I mentioned in the beginning, we are able to use this paper device to enrich and purify the specific ion genetic materials from sewage. So that's avoids the interference from the other impurities in sewage. So secondly, we have a very specific molecular assay. It's called LAMP. So that is a very sensitive and selective detection of the genetic target, which ensure that those detections should be very selective and sensitive. Thirdly, we also have a control to ensure that the test is valid in terms of the reagents to avoid the false positive and the false negative signals. So we are also build up our confidence from our previous experience by testing other organisms such as malaria and leptospirus and brucellus. So it works well so far.
Do you have a control specifically that is COVID-19, like a pure isolated virus sample?
So in terms of the real experiment, we have the plasmid is artificial genetic sequence as a positive control to validate our test. Yeah, it will be also integrated with our device.
So you artificially replicated like something that resembles COVID-19.
Yeah, the specific sequence. Yeah, this is also, I think many people using this method because to access of the real viral sample is very difficult for most of the labs. They need to get the P3 level health and safety.
Maybe I should put you into contact with Dr. Tasnim Soliman, because this is recently now that the news has been published that she has here in South Africa isolated a pure sample of coronavirus. And if you require perhaps like a more like pure sample that's actually isolated COVID-19, perhaps I can put you into contact with her and then you can maybe use that sample. I'm not exactly sure how the transport system will work for that. But if you'd like to maybe try to, I'll send you the details for her.
Okay, that should be very useful.
Yeah, they have a BSL-3 lab here and they established a way of isolating the virus and making a pure sample of it.
Yeah, you mean the isolate, the culture, the coronavirus sample from sewage?
Yes.
Or from, okay.
I think they,
That's interesting.
I'm not sure
that's very
exactly.
useful.
Yeah, I should ask the details of how they managed to do it. But
Mm-hmm.
the news was very just more like the practical aspect from this is what has happened. It didn't really
Yeah,
go into
yeah,
detail of exactly how
yeah,
it was cultivated. But yeah, I'll put you two into contact. Maybe I can help you with the
yeah, yeah, yeah,
further progress of establishing some sort of control and avoiding false positives. But that's also the way
yeah,
you're doing it now, avoiding false positives just by testing with other known viruses that you know that
yeah,
that's
yeah, yeah. It's a
yeah
method of validation. So we
yeah
are
yeah
sure that we can just make sure that there is no false positive or no false negative signals.
yes
This is very important for the device performance. Yeah.
yeah. Said you tested COVID-19 with the device. You've used that now already, right? How long does the
Not really. It's still
still prototype
under
and okay,
development
but
because I
in
have to be
the
honest that yeah, the university lab was closed for 6 weeks, so
yes
we can't do
no the
have any
same
lab activity. So yeah,
no yeah, so nothing's really coming out of the lab right now.
yeah, yeah.
um
But we unfortunately we just get access. Now we can carry out the further
yeah,
experiment.
that's
Yeah,
great. Yeah, because it's a priority. That's great.
yeah.
But in your other tests for, let's say, for malaria, how long does the results take for you to see a positive result or to
So
see
it
a negative?
really takes 1 hour
One
from
hour.
sample to answer. Yeah,
Okay. Like
less than
PCR
one
usually
hour.
takes like 6 hours to get a result. So
Yeah,
that's
yeah,
also very good because now you can compare it with like you can get information much faster.
yeah. Thank you.
Um, we've run through all the questions that I wanted to ask you. I'm so glad you gave me all the information that I need. Okay, the last thing I wanted to ask you was just from like a business standpoint, how much does this device cost and the manufacturing and development process of it cost? Would it be possible to make this in like a very large scale? You know, how
Yeah.
does that look?
Yes, I think this device, the cost is very low, as mentioned in the beginning. First,
Okay,
the materials from the paper, which is very cheap materials,
yeah,
and
it's
we
made
have
from
the
cellulose mostly, right?
sorry.
Is it made from cellulose
Yes,
mostly?
cellulose paper. Yes,
Yes.
that's true.
Okay.
Chromatography cellulose
Yeah.
paper. So have the rough estimation. The single device will cost less than £1 roughly, but
All
it's
right.
obviously involve our intellectual properties and all the technology development. So that's our all material cost in terms of the manufacturing. And go back to your second question, it's definitely possible to do a large scale manufacturing in order to promote and be used for other people. So we are still working to improve that to make it more user friendly, which means that even a non-expertise can manipulate this assay by themselves. So we hope that this device can be widely used in order to, as a rapid test to check if any infections enter the community.
Yes.
Yeah, that's our ambitions as well.
Yeah. No, but it sounds very, very promising, and because it's not renewable, but it's environmentally friendly. It's relatively cheap to make, and it's very easy to transport and use everywhere and by almost anyone. You don't have to be terribly skilled to be able to understand or use it. It might have a very good promise for the future, not just for COVID-19 testing, but just for any other type of pandemic that might occur in the future to establish some sort of, easy testing, bulk testing type of device. That's very, very great. I'm very excited about this project.
Yes, thank you.
Yeah, thank you for your time and thank you for like sharing the information with me and with everybody who's going to be seeing this.
Thank you. It's my great pleasure. That's very good. So, by the way, I also get a PhD in polymer science.
Oh,
I work on polymer science or chemistry.
yes, yeah. Yeah.
What topic are you working on polymer
Oh,
science?
I finished my PhD last year, but I worked specifically also, I was also interested in removing or cleaning treatment of wastewater, but I ended up going more into the removal of heavy metals from wastewater. So I
Okay,
developed a sponge and worked with chitosan to find like an environmentally friendly adsorbent that can help remove heavy metals from wastewater. So
okay.
My project
That's
mainly
a
revolved around
rural.
developing the materials for the adsorbent, but it was a very, very interesting and fun project for me. So I like this topic a lot.
Yeah, the reason I ask is for my first degree and master degree, I also working in polymer science and
Oh, yes.
engineering. So I also working in some polymer materials. So that I know a little bit also like Kytogen. I want very good biocompatible materials. And I did do some research in terms of polymer mechanical properties of polymer material, like crystallization and the mechanical properties.
Yeah.
So that's good as we are in more or less very close areas.
Yeah. No, it's
Yeah.
amazing actually how all the fields just like combine into one, and I'm really like polymer science, especially. I feel you can branch into so many aspects of polymer science, you know. But thank you so much for the interview. I really appreciate it. It was so much fun speaking to you.
Thank you.
Enjoy your
Okay.
day.
See you later.
Goodbye.
Thank you. You too.